Today we're speaking with Dr. Joan Massagué, Chairman of the Cancer Biology and Genetics Program at Memorial Sloan-Kettering Cancer Center and an AACR board member. He presented the talk, Microenvironment Survival Signals for Disseminated Cancer Cells. Thank you for joining us.
You're welcome. Pleasure.
What are some of the ways we can identify survival mechanisms in circulating tumor cells?
Well, circulating tumor cells are of great interest nowadays. It's understood that cancer is a systemic disease, and there is the primary tumor that releases cells even from way before we have diagnosed it and, of course, removed it. And then there are the metastases that may not be growing for some time, but are seeded already. In between the two, of course, there are cancer cells that are circulating from one to the other. There is a great deal of interest in capturing these cells and in interrogating them. However, this is still technically very tricky. Cells, circulating tumor cells, can be isolated with a number of technologies, but they are still rudimentary, these technologies, in terms of providing us investigators with cells that can be interrogated for what is that they are doing at that point, how healthy they are these the most aggressive cells in the population, or the least, or what. Therefore, in order to approximate the question, we have to act by inference, by studying what is that the end result of the circulation of these cells is. For example, what is the load of seeding that exists in various organs? How does that relate to the robustness with which the primary tumor has been releasing these cells? How are these cells going to relate to disseminated cells to the outcome that these patients endure? That is, whether they develop metastases aggressively and in what organs. So we are approximating the problem and the answer to these questions through inference, through triangulation, while we wait with great interest for the technologies of circulating tumors, isolating circulating tumor cells to evolve to the point where they will provide us with entities for investigation in the lab.
Would you discuss your research identifying metastasis progression genes and pathways that are responsible for breast cancer metastasizing into the bones and lungs?
Certainly. So we consider that metastasis is the end product of an evolution process that has three fundamental phases. The phase of dispersion. Cells, cancer cells, leaving the tumor, passing to the circulation directly or via lymphatics, and then resisting that and extravasating, infiltrating, passing from the bloodstream into tissue. That's the first phase. But it has all these steps already, and each one of them requires and selects for and demands of the cancer cells certain abilities provided by genes and pathways that may be active or not. The second phase is what they do on arrival in a given organ. How do they survive the shock of an environment that they have never been accustomed to? And how do they get to keep the tumor initiating state or stem cell ability that they will have to resort to in order to recommence a tumor? That's the second phase. The third phase is the recommencement of an aggressively growing metastasis, what we call the metastasis, the one that is detected, that is painful, that shows up in an image, sometimes years, sometimes months, sometimes decades later. Each one of these three phases is a compendium of biological processes. So we approximate these by two approaches. One is to interrogate the end products, isolate from patients with advanced disease cells, cancer cells that have graduated through the entire process and are highly aggressive to the bones or to the lungs or to the brain. Of course, they have evolved that way by adapting to one or another of these organs. So they are somewhat different, much like different finches in different Galapagos Islands. So we can interrogate these cells, compare them to one another, and ask what is that the ones that are nesting very efficiently in the bones have that the ones that fail to do that, maybe they are very good in the lungs, do not have. And we study this by asking cells from a patient to perform these functions in an experimental animal model, the mouse. We also approximate these by interrogating the source tumors. We ask what is that tumors have that that allowed certain tumors in a certain subset of patients to seed that patient with cells that were, for example, capable of resisting for a long time in the bone marrow or somewhere, that allowed them 20 years later to outgrow us as bone metastases. They were not very good at the beginning at growing as bone metastases, but they were certainly had to be very, very good at resisting and staying alive for all that time. So by interrogating both the source tumor and the end products, and then doing archaeology back and forth, we extract information about candidate genes, candidate pathways, and candidate processes that underwrite the ability of these cells to fulfill the properties in each one of these three major phases or chapters of the metastasis process.
Would you expound on the potential of this research to open new avenues for treatment of metastatic cancer?
Certainly. We need knowledge. We need to know how metastasis runs in its very many variants, depending on the tumor type, depending on the metastasis site, depending on the course of the disease, with long periods of latency or very quickly metastatic in most cases by the time the primary tumor is diagnosed. We need to put all this information on the table, understanding that out of maybe 20 discoveries that we are going to make, only one will be actionable with regards to therapy. Many more will be actionable with regards to diagnosis and prognostication of who is at risk of what. Then, out of this mass of information, like I say, there will be that one finding, that one molecule, that one target that is very promising, it's addressable. And then we have to think about what phase do we attack in metastasis? We, of course, react with intensity to wanting to do something about the overt metastatic growth, that brain metastasis nodule that is giving somebody neurological symptoms, that lung metastasis that shows up, that bone metastasis. Of course, we must do as much as we can, but we can do better than this by preventing it. That is, by understanding the biology of that disseminated disease that is seeded, that is invisible, but are the seeds of the metastasis that will manifest itself months, years, sometimes decades later. So if I put my money, so to speak, on something, is in this, identifying the basis for the survival and fitness of latent disseminated disease, attacking that, and with that, preventing metastases from showing up in the first place.
Dr. Massagué, thank you so much.
Thank you.